This is a representation of a cell before it begins meiosis, a process in the nucleus that divides the chromosome number in half. For clarity, the nuclear membrane is not shown. Also, the chromosomes are depicted as condensed, although during interphase of the normal cell cycle, they are actually thin and dispersed and not visible under a light microscope. Before a cell enters meiosis, it first replicates its DNA. After DNA replication, the chromosomes are duplicated so that each has two identical sister chromatids connected at a structure called the centromere. Meiosis consists of two successive cell divisions called meiosis I and meiosis II, each of which is subdivided into 4 phases. In the first phase of meiosis I, called prophase I, the homologous chromosomes, or homologs, in a diploid cell come together in a process called synapsis. The synapsed pairs are known as tetrads. Each tetrad consists of one chromosome inherited from the mother, colored red in the diagram, and one inherited from the father, colored blue. Where they're synapsed, the chromosomes can cross over each other, forming an X-shaped structure called a chiasma. At the crossover sites, the homologous chromosomes exchange segments. This exchange results in genetic variability in the daughter cells. The second major phase of meiosis I is metaphase I. During this phase, the pairs of chromosomes line up in the middle of the cell. During the third phase, anaphase I, the chromosomes of each pair move to opposite poles of the cell. In telophase I, the fourth and final phase, the chromosomes reach the poles of the cell. When meiosis I is complete, the cytoplasm divides to produce 2 haploid daughter cells, each having just a single set of chromosomes. Even though a duplicated chromosome has 2 chromatids, the chromatids are bound by a centromere and are considered one chromosome. The number of chromosomes in a cell equals the number of centromeres present. Note that the 2 daughter cells are genetically different. The second event of meiosis, meiosis II, strongly resembles mitosis. During prophase II, duplicated chromosomes, consisting of 2 sister chromatids, begin to move to the middle of the cell. In metaphase II, the chromosomes are arranged along the cell's midplane. The sister chromatids begin to separate in anaphase II, becoming independent chromosomes that move to opposite poles of the cell. During telophase II, the chromosomes reach the poles. When meiosis II is complete, the cytoplasm of each cell divides to form 2 daughter cells. The 4 cells are haploid, each containing a single set of chromosomes. Note that the 4 daughter cells are all genetically different from one another. A cell that undergoes mitosis produces daughter cells that are dramatically different from the daughter cells produced by a cell that undergoes meiosis. Let's compare these 2 processes, beginning with a look at mitosis and meiosis I. In both mitosis and meiosis I, cells begin with replicated chromosomes, each cell having 2 chromatids per chromosome. In meiosis I, the homologous chromosomes pair with each other and come to the middle of the cell together. In mitosis, the homologs move independently of each other. In mitosis, the chromatids separate. In meiosis I, the homologs separate. In mitosis, a diploid cell divides to produce genetically identical diploid cells. Each cell contains 2 complete sets of chromosomes. In meiosis I, a diploid cell divides to produce genetically unique haploid cells. Each daughter cell now contains just one set of chromosomes. Meiosis I is known as a reduction division because the outcome is a reduction of chromosome number. These 2 cells are daughter cells produced from meiosis I. Let's use just one of them to compare meiosis II to mitosis. A cell entering mitosis is diploid, with 2 complete sets of chromosomes. A cell entering meiosis II is haploid, with just one complete set of chromosomes. In both cells, each chromosome has 2 chromatids. In both mitosis and meiosis II, the chromosomes migrate to the middle of the cell. Then, the sister chromatids of each chromosome separate to form independent chromosomes. In mitosis, a diploid cell divides to produce genetically identical diploid cells, each with 2 complete sets of chromosomes. In meiosis II, a haploid cell divides to produce genetically unique haploid cells, each with 1 complete set of chromosomes. If we consider that meiosis II actually began with 2 cells, then the result is 4 genetically unique haploid cells. In both mitosis and meiosis II, the division is not a reduction division. Only in meiosis I are the number of chromosome sets divided by half.